anatomy. For this anatomy masterclass webinar series, today Dr. Sandeep, let me share first of all. Is my screen visible?
Yes, ma'am. Yes, ma'am.
Yes.
So today, Dr. Sandeep Pakhale sir is going to deliver lecture on development of brain. In the last session, Dr. Yogesh Sontakke sir, he explained about the development of spinal cord. Dr. Gagnesh sir, we have listened, he has explained about development of heart. So Sandeep Pakhale sir, before we start, let me introduce sir. Sir is currently working as a professor in the Department of Anatomy, Dr. Balasaheb Vikhe Patil Rural Medical College, Loni, that is Pravra Institute of Medical Sciences. Sir is PG and PhD guide. Sir is also member of Medical Education Unit. Sir is also director, IQAC cell. Sir has more than 19 publications. Sir has received Best Poster Award, ACME. Sir also received Best Research Award in Anatomy Regional Award. Currently, Sir is member of Board of Studies, MGM Institute of Health Sciences, Navi Mumbai. Sir is former BOS member at KVV Karad. Sir is also former Executive Committee member of Regional Chapter of Anatomy. With this, I welcome Dr. Sandeep Pakhale, sir.
Thank you,
Now,
madam.
screen is handed over to Pakhale, sir. Those who have any doubts, queries, can write in chat box. They will be solved at the end of class. Dr. Sandeep Pakhale, sir.
Is my screen is visible?
Yes, sir.
Thank you. Good evening, respected senior faculty members, distinguished colleagues, office bearers of Regional Chapter of Anatomy, and dear postgraduate students. I would like to begin by acknowledging the excellent session by Dr. Shingare sir on leadership in anatomy, also by Dr. Yogesh Sontakke sir on the development of spinal cord and its anomalies. The way he integrated embryological events with the clinical relevance provides a perfect platform for what we will discuss next. First of all, I would like to greet from Pravra Institute of Medical Sciences, Loni. I would like to express my sincere gratitude to the office bearers of Regional Chapter of Anatomy, President Dr. Prafulla Dattwe sir, Vice President Dr. Shilpa Gosavi ma'am, Secretary Dr. Rakhi More ma'am, Editor-in-Chief Dr. Praveen Iyer sir, and all Executive Committee members, and the moderator for this session, Kirti madam. Continuing along the neuraxis, we now move from the relatively linear organization of the spinal cord to the far more complex and dynamic process of development of brain. Unlike spinal cord, brain development is not just about elongation and segmentation. It involves cephalization, vesicular differentiation, flexural changes, and differential growth, all of which occur in a tightly regulated temporal sequence. For a postgraduate, the real value lies not just in recalling stages, but in understanding mechanisms. These are the competencies. Now, to recap the previous session, how there is a beginning of neural development starts. What we are seeing here is the transformation of ectoderm into a neural plate, which we usually say slipper-shaped neural plate, under the inductive influence of notochord. So you can see here notochord. The neural plate starts to invaginate from the neural groove with elevation of neural folds on either side. So the neural crest originate from the margins of the neural folds during neural tube closure. They undergo an epithelial mesenchymal transition. They detach and then migrate extensively along the dorsolateral and ventral pathways. What is important to appreciate is their multipotency. They contribute not only to the peripheral and autonomous nervous system, also dorsal root and sympathetic ganglia, but also to the structures like adrenal medulla. Clinically, any defect in their migration or differentiation leads to a group of disorder called as neurocristopathies, which includes Hirschsprung and neuroblastoma. At this stage, the neural folds elevate and begin to fuse, converting the neural plate into a neural tube. So this is, you can see the two diagrams here, day 22 and day 23, wherein closure has not occurred and both the closures does not occur simultaneously. It starts in the cervical region and then progresses both cranially and caudally. For a short period, the tube remains open at both ends as cranial and caudal neuropores, wherein you can see this is the cranial neuropore and this is the caudal neuropore. The cranial neuropore closes at about 25th day, 25th day, followed by caudal neuropore at around 27th day. Clinically, if there is a failure of closure at these ends leads to neural tube defect that already we have seen. Now coming to further development. So here we are looking at the earliest stage of development. The portion of the neural tube cranial to fourth somite. So you can see here, cranial to fourth somite leads to expansion and form the brain. We can identify three dilatations, which are primary brain vesicles, in which first one is prosencephalon, which is also called as forebrain. Second one is mesencephalon, which later on forms midbrain. And the third one, that is hindbrain or rhombencephalon, which further forms the hindbrain. An important landmark to remember is that the site of closure of anterior neuropore, which forms the lamina terminalis. So at this stage, the brain is essentially organized into three primary vesicles. And already I have told you prosencephalon, mesencephalon, and rhombencephalon. So these are called as primary brain vesicles. Now, as development progresses, each of the primary vesicle undergoes further differentiation, starting with forebrain. The lateral walls show evagination forming the optic vesicles. And from these three primary vesicles, 5 secondary vesicles are formed. Slightly later, additional outgrowths give rise to the cerebral hemisphere. And this leads to subdivision into telencephalon, which forms the hemispheres and lateral ventricles, and the diencephalon, which further forms the thalamus, hypothalamus, pituitary gland, and the cavity of the diencephalon is third ventricle. The cavity persists as the third ventricle, and it is connected to the lateral ventricle via foramen of Monro. So these are primary vesicles which are started converting into the further 5 vesicles. Here, the midbrain remains relatively unchanged externally, but its cavity becomes narrow to form the cerebral aqueduct. So from outside, it remains unchanged, and this cavity will form the cerebral aqueduct, an important CSF pathway. The hindbrain divides into meten and myelencephalon. So this part is divided into two parts, meten and myelencephalon, forming medulla oblongata. And the cavity of this is fourth ventricle. Fourth ventricle. The wall differentiates into ependymal, mantle, and marginal zones, as we have seen already in the development of spinal cord. And it is divided by the sulcus limitans into ventral motor, which is also called as basal plate, and dorsal sensory, which is called as alar plate. So you can see the telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So these are 5 secondary vesicles: telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So these are called as 5 secondary brain vesicles. This diagram, as we are seeing, summarizes the transition from secondary brain vesicles to adult derivatives. Starting at the top, the telencephalon, so this part here, it expands massively to form the cerebral hemispheres. So this is forming the cerebral hemispheres, which dominate the adult brain. Just below it, diencephalon, this yellow colored portion, this gives rise to keep deep structures such as thalamus and hypothalamus. So this portion is thalamus and hypothalamus. The mesencephalon or midbrain, it remains relatively conserved. So this is midbrain. Midbrain. Further caudally, the metencephalon develops into the pons and cerebellum. This metencephalon is divided into two parts, that is pons and cerebellum. So what is important to appreciate is that each secondary vesicle has a direct and predictable adult derivative, which helps in understanding both anatomy and clinical localization. This metencephalon will form medulla oblongata, and lower part of this is neural tube is spinal cord, which already we have seen. So telencephalon gives rise to cerebral hemisphere, diencephalon thalamus hypothalamus, mesencephalon midbrain, pons cerebellum, medulla oblongata. Now coming to the cavities of these. Now let us look at the cavities of the developing brain, which are essentially modifications of the lumen of the neural tube. The lumen of the spinal cord persists as the central canal that we have seen, and this cavity continues throughout the brain. So this cavity is continuing throughout the brain. As development proceeds, each vesicle forms a specific ventricular component. The rhombencephalon, which gives rise to fourth ventricle. So this portion is giving rise to fourth ventricle, which lies between the brainstem and the cerebellum. Other cavities are telencephalon is having lateral ventricles. So these are lateral ventricles. There are two lateral ventricles. The lumen of the diencephalon forms the third ventricle. This is third ventricle, which is midline cavity and associated with thalamic structures. The telencephalon with expanding cerebral hemispheres has divided into two parts, and two lateral ventricles are formed, which are communicating with each other with the help of foramen of Monro. Foramen of Monro. And primarily, the midbrain cavity narrows. So this cavity narrows to form the cerebral aqueduct. Cerebral aqueduct. So you can see cerebral aqueduct, fourth ventricle, which is the cavity of the hindbrain, fourth ventricle. So upper part and lower part. Upper part is open, which is considered as cavity of the metencephalon, pons and cerebellum, while myelencephalon, lower part of the fourth ventricle. So now, folding of the neural tube. In this slide, we can see folding of the neural tube, which is responsible for the characteristic shape of the brain. And these foldings forming the flexures of the brain. First is mesencephalic flexure. So this is mesencephalic flexure. The first flexure to appear is mesencephalic flexure around fourth week of intrauterine life at the level of midbrain. So this is at the midbrain level. It is ventral flexure. It is ventral. So the concavity is on the ventral side. Next is the cervical flexure. Cervical flexure. So you can see this is a cervical flexure. This appears around fifth week of intrauterine life at the junction of hindbrain and spinal cord. So this is spinal cord and this is hindbrain part. And at that level, this cervical flexure is formed, which is also concave ventrally. This also bends ventrally and helps to demarcate the myelencephalon from the spinal cord. However, it gradually reduces and disappears as the development proceeds. So you can see first to begin with it is, and slowly it is reducing. Now the third flexure is pontine flexure. And you can see this is the pontine flexure. And this is concave dorsally. It separates the metencephalon from the myelencephalon. This pontine flexure is particularly important because it causes opening out of the hindbrain and leading to formation of rhomboid shaped fourth ventricle and rearrangement of the alar and basal plates. So overall, these flexures not only change the shape of the brain, but also influence its internal organization and ventricular anatomy. So these are folding of the embryo, and you can see neural tube. We can see the cephalic flexure here, ventral side. This is ventral, this is dorsal, this is pontine flexure, and this is cervical flexure. Now focusing on the hindbrain or rhombencephalon, it is divided into two parts. The myelencephalon, which is the most caudal part and continues inferiorly with the spinal cord, and eventually forming the medulla oblongata. So medulla oblongata is formed as a myelencephalon. Now coming to the formation of medulla oblongata. Above this lies metencephalon, which extends from the pontine flexure up to the rhombencephalic isthmus, the junction between midbrain and this one. So you can see here the metencephalon gives rise to pons and cerebellum, while the myelencephalon forms the medulla oblongata. So structurally, the hindbrain is organized between the pontine flexure below and the isthmus above. And functionally, it contributes to the vital brainstem and cerebral components. Now, focusing on the myelencephalon, which forms the medulla oblongata, unlike the spinal cord, the key change here is due to the pontine flexure, which causes the lateral wall to evert or open out. And how it is opening, that we will see. Because of this, the cavity expands. So cavity is expanding and forming the fourth ventricle, giving rhomboid shape. So in spinal cord also, we have seen there is a floor plate, there is a roof plate, and the two other plates are alar plate and the basal plate. So roof plate is there, and roof plate is stretched laterally like opening a book. So this will be later on opening like a book, like this. So this eversion also leads to an important rearrangement in which the alar plate, which are sensory, they shift laterally. So this is alar plate, they shift laterally. While the basal plate, they remain medially. They remain medially. So alar plate is responsible for sensory function and the basal plate is responsible for the motor function. So compared to the spinal cord, where sensory is dorsal and motor is ventral, in the medulla, we see medial is motor and lateral is sensory organization. The basal plate forms the motor nuclei. So you can see here, this portion will further later on form the motor nuclei, while the alar plate will form the sensory nuclei on both the sides. So even in brainstem, the columnar organization of motor function is preserved. So you can see here, Here you can see sensory, from the sensory part, some of the nuclei, they migrate towards ventrally and medially. And these are olivonucleus from the alar plate. An important feature in development of medulla is migration of the olivary nucleus. This migration, neurons of the inferior olivary nucleus originate in the region, basal plate region of the myelencephalon due to effect of the pontine flexure, the eversion of the lateral walls. These neurons undergo ventrolateral migration. As a result, instead of remaining in the medial position like typical motor nuclei, the olivary nucleus comes to lie laterally, forming the characteristic olive on the surface of the medulla. Functionally, this nucleus establishes connection with the cerebellum, playing a key role in the motor coordination. So this is inferior olivary nucleus. Olivary nucleus. Motor coordination and learning. The alar plate forms sensory columns. We have already seen alar plate is responsible for the sensory function. Here, the sensory columns which are arranged from medial to lateral, are general visceral afferent, spatial visceral afferent, spatial somatic afferent, and general somatic afferent. So details of all these functional components in the alar plate we are going to see. The basal plate forms motor columns which are arranged from medial to lateral, and that is general somatic efferent. General somatic efferent, that is medial one. The intermediate one, this one, intermediate one, spatial visceral efferent, and lateral is general visceral efferent. So these are motor functions or motor columns. In the myelencephalon, the roof plate now. So already two portions we have seen. So this is motor, this is sensory functional component, and this is roof plate. This roof plate is thin, consisting of single layer of ependymal cells covered by vascular pia mater, and together they form telachoroidea. They form telachoroidea. Due to vascular proliferation, this region forms the tuft of capillaries or tuft-like invaginations into the ventricular cavity. So this is ventricular cavity, which develop into choroid plexus. And this choroid plexus is responsible for CSF production. Now coming to metencephalon, development and derivatives. The metencephalon, like the myelencephalon, is organized into, again, basal, motor, and alar sensory plates. From alar plates, the cerebellum develops, which plays a key role in coordination of the movement and also the posture. Coordination and posture. While the basal plate, along with marginal layer, they are responsible for forming the pons and acting as a major conduit for fibers connecting cerebellum, and spinal cord. The pons contains the pontine nuclei, which is derived from the alar part. The pons develops from the metencephalon, and an important event here is the ventral migration of pontine nuclei. So you can see there is a ventral migration of the pontine nuclei. These nuclei originate from the alar plate. They are originating from the alar plate of the both metencephalon and myelencephalon and migrate ventrally to occupy the final position in the pons. At the same time, the marginal layer of the basal plate expands, forming the transverse fiber tracts, giving the pons its characteristic role, which we say bridge between the cerebral cortex, cerebellum, and spinal cord. So that's why pons means bridge. So the pons is a combination of migratory sensory derived nuclei and major conducting pathways. In the pons, the basal plate forms the motor nuclei. Basal plate forms the motor nuclei, including those of the trigeminal, abducens, and facial nerves. The alar plate forms the sensory nuclei, particularly for trigeminal and facial components, and also gives rise to the pontine nuclei, which are important relay centers. So the basic rule still holds: basal is motor and alar is sensory, with the added contribution of the pontine nuclei from the alar plate. The basal plate forms motor nuclei, which are arranged as we saw, like in medulla, from medial to lateral. These include somatic, branchial, and visceral efferent groups. So general somatic efferent, branchial efferent or spatial visceral efferent, and general visceral efferent. And these include abducent nerve nuclei, trigeminal nerve nuclei, and facial nerve nuclei, along with secretomotor components. The alar plate forms sensory nuclei, again arranged lateral to medial pattern, that is general somatic afferent, spatial visceral afferent, and general visceral afferent. So if we see here, there is a column theory, and we can see the closed part of medulla, open part of medulla, pons, and midbrain, which we are going to see the development, in which from medial to lateral, that is, these three are basal and these four are alar plates. Here is a summary of the functional columns of the basal and alar laminae in the brainstem. Starting with the basal lamina, which is motor, the columns are arranged from medial to lateral as somatic efferent, which is supplying muscles derived from pharyngeal arches. So you can see here the hypoglossal nerve nucleus, abducens nerve nucleus, trochlear nerve nucleus, and oculomotor nerve nucleus, and supplying the extraocular muscles and the muscles of the hypoglossal, that is tongue. The second column is spatial visceral efferent and supplying the muscles derived from pharyngeal arches again, that is nucleus ambiguus. So supplying smooth muscles and this general visceral efferent, they are supplying to the smooth muscles and the gland. So you can see here 12th, 11th, 7th, and 5th. These nerves, that is spatial visceral efferent or branchial efferent, we can see. The general visceral efferent they are supplying to the dorsal nucleus of vagus, Edinger-Westphal nucleus. So that is general visceral efferent. They supply the smooth muscles and the glands. Moving to alar lamina, which is sensory from medial to lateral, supplying the arrangement is essentially reserved general visceral afferent carrying interoceptive input. So this is general visceral efferent. So nucleus of vagus, that is nucleus of 7th and 9th glossopharyngeal. That is general visceral afferent. Next, lateral to that, there is a spatial visceral afferent, mainly carries the taste sensation. Taste sensation. And the most laterally, we have spatial somatic afferent, which is responsible for hearing and balance. Hearing and balance. So this is how from medial to lateral, these columns, motor and sensory columns, are lying in the brainstem, we can say. So this table summarizes the functional cranial nerve columns. The motor column includes general somatic efferent, which is responsible for somatic striated muscle, that is extrinsic muscles of the or extraocular muscles of the eye and muscles of the tongue. And the nerves involved are oculomotor, trochlear, abducent, and hypoglossal. And these nuclei, they lie in myelin and metencephalon. The second one is spatial visceral efferent or branchial efferent, responsible for striated muscles of the pharynx. So muscles of the branchial apparatus. These are the muscles derived from the pharyngeal arches. Again, the nerves involved are trigeminal, facial, glossopharyngeal, and vagus. And these nuclei lie, as we saw in the previous diagram, these locations are metencephalon and myelencephalon. The general visceral efferent, which is responsible for parasympathetic pathways of the eye and smooth muscle, that is sphincter pupillae, airways, viscera, heart, and salivary glands. The nerves involved are again 3rd, 9th, and 10th. So these are all motor functional component. Now coming to the fourth, again from medial to lateral, sensory component, general visceral afferent. So they carry the visceral sensations from the GI tract, and the nerve responsible for it is vagus nerve. The nucleus lies in the myelencephalon. Then lateral to that, there is a spatial afferent, spatial visceral afferent. And this is for the taste sensation and taste from the tongue, palate, epiglottis. Nerves carrying these tastes are the facial nerve and the glossopharyngeal nerve. Lateral to that, we have spatial afferent, that is spatial somatic afferent, which carries the hearing and balance sensation, and structures innervated by the vestibulocochlear nerve, that is cochlea and semicircular canals. Again, it lies, location of the nerve functional nucleus is metencephalon. So this table integrate function, innervation, and cranial nerve distribution in a columnar pattern. This diagram shows lateral view of the brain vesicle in an 8-week embryo where the basic organization of the brainstem is now well established. The roof plate of the rhombencephalon has been removed. So you can see here the roof plate. This is the cut edge of the roof of the rhombencephalon. And this allows us to visualize the intraventricular portion of the rhombic loop, which contributes to cerebellar development. At this stage, the effects of pontine flexure are evident with hindbrain opened and the fourth ventricle is clearly formed. Importantly, we can also appreciate the origin of the cranial nerves, which are arranged according to functional columns, which already we saw in the last two slides, derived from basal and alar plates. Now coming to the development of the cerebellum develops from the alar plates of the metencephalon. Alar plates of the metencephalon, highlighting its sensory origin. So you can see from this cut edge, the cerebellum is formed. Specifically, it arises from dorsolateral thickening called as rhombic leaf. So this is called as this dorsolateral rhombic leaf. These rhombic leaves grows medially and eventually fuse in the midline. So they grow medially and eventually fuse in the midline, and they form the cerebellar primordial. So the key concept is that the cerebellum originates from alar plate via rhombic leaves and later undergoes extensive growth and organization that we will see. The rhombic leaves, you can see here, they are growing medially and fusing at the cerebellar plate. Specifically, it arises from the dorsolateral thickening, which I shown you in the previous diagram also. As the development progresses, the rhombic leaf grows medially towards each other and eventually fuse in the midline, forming the cerebellar plate. So this is cerebellar plate. This is cerebellar plate. This cerebellar plate, then differentiates into two main parts. The midline portion is called as vermis, and the lateral vermis. The vermis is primarily involved in the axial coordination, while the lateral two portions, they are called as cerebellar hemispheres. So this portion is called as cerebral hemisphere, and this midline portion is called as vermis. The lateral cerebellar hemispheres contribute to the fine motor control. Fine motor control. The vermis is primarily involved in axial coordination. From a simple pair of lateral thickenings, we get a midline structure and bilateral hemispheres, forming the basic architecture of the cerebellum. So midline is vermis and two lateral plates. With further development, extensive cell migration leads to the formation of characteristic three-layered cerebellar cortex. And these three layers we all know: molecular layer, Purkinje cell layer, and granular layer. These layers includes, this is outer one, outer molecular layer, middle Purkinje cell layer, and inner granular layer. The Purkinje cells act as the principal output neuron, while the granular layer contains densely packed interneurons. As the cerebellum grows rapidly, its surface undergo folding. So you can see here, it is growing, but there is no space to expand it. So it gets folded like this. So it gets folded like this. So due to this folding, the surface area is increased and it enhances the functional capacity. In the latter stage, the cerebral development cells from the external granular layer, they migrate inward. So you can see here, this is external granular layer and they start migrating inward. These cells interact with the Purkinje neuron. So this dotted strip, or you can say these are the Purkinje neurons, and contribute to the formation of definitive cerebellar cortex, establishing its mature layered architecture. Within the cerebellum, we have the dentate nucleus, which develops as one of the major deep cerebellar nuclei and playing an important role in motor coordination. Also, you can see presence of anterior and posterior medullary vellum or vela, which are thin structures related to the roof of the fourth ventricle. So this stage represents the final organization of the cerebellar cortex and deep nuclei. By the sixth month, that is late stage, the external granular layer becomes highly active and gives rise to multiple neuronal types. So multiple cell types are formed at the sixth month. These cells migrate inward, forming granule cells, which are interacting with the Purkinje cells, contributing to the cortical organization. At the same time, basket cells and the stellate cells, they arise, and they are arising from proliferating cells within the cerebellar white matter. The cerebellar cortex, which is composed of Purkinje cells, Golgi type 2 neurons, and granule cells. They continue to mature and reaches the definitive size after the birth. The definitive cortical size is achieved only after birth. In contrast, the deep cerebellar nuclei, such as dentate nucleus, attain their final position before birth. So that is the only thing which attain the maturity before birth, that is dentate nuclei. In the prenatal stage, the external granular layer forms a proliferative zone. It forms the proliferative zone on the cerebellar surface. So outermost external granular floor, that is proliferative zone, giving rise to granule cells and migrate inwards towards the Purkinje layer that we saw. At the same time, basket and stellate cell develop from the cerebellar white matter. Postnatally, that is after birth, a well-defined three-layer structure is formed with outer molecular middle Purkinje cell layer and inner internal granular layer. So the cerebellum transitions from surface proliferation to inward migration, ultimately forming a mature layered cortex after birth. Now coming to the formation of the midbrain or the mesencephalon. This region represents the least modified part of the developing brain, yet it plays a crucial role. In the next few slides, we will focus on how the basal and alar plates differentiate, giving rise to important structures like crus cerebri and colliculi, and how this relates to the clinical and functional anatomy. The marginal layer of the basal plate enlarges to form crus cerebri. So this marginal layer enlarges to form the crus cerebri. In the mesencephalon, the basal plate forms the motor nuclei, like we have seen in pons, medulla, spinal cord. So this basal plate forms the motor nuclei. Again, already we have discussed this column theory, in that from medial to lateral, the motor nuclei are oculomotor nerve nucleus and trochlear nerve nucleus, which lies in the midbrain, supplying to the extraocular muscles. Lateral to that, we have general visceral efferent Edinger-Westphal nucleus supplying to the sphincter pupillae, and the marginal layer, that is crus cerebri, descending cortical fibers to the brainstem and spinal cord. So each basal plate differentiate into somatic efferent and visceral efferent nuclei. It includes the oculomotor and trochlear nerves, which supply the extraocular muscles. In the midbrain, the basal plate gives rise to motor nuclei. So you can see here, this is somatic efferent and this is visceral efferent. Somatic efferent, oculomotor and trochlear, and the visceral efferent is Edinger-Westphal nucleus. So this is Edinger-Westphal nucleus, and this is oculomotor nerve nucleus and trochlear nucleus. The marginal layer of the basal plate expands to form the crus cerebri. So you can see here, this is expanding to form the crus cerebri, which carries the descending fibers from cerebral cortex to the lower centers, and they are passing through the crus cerebri. On the other hand, the alar plate forms the tectum. So you can see there is a migration of this alar plate which develops into the, this is migration, this leads to form first tectum, and which includes the superior and inferior colliculi. The superior colliculi are involved for visual reflexes, and the inferior colliculi serves as a relay center for the auditory reflexes. So you can see this migration takes place in the tectum. In the midbrain, the alar plate forms the sensory component. So this portion, they form the sensory component, specifically the tectum. And you all know that there are superior and inferior colliculi. The tectum is divided into superior and inferior colliculi. The superior colliculi acts as the center for visual reflexes, coordinating eye and head movements in response to the visual stimuli. The inferior colliculi function as a auditory relay and reflex center, possessing sound-related inputs. So auditory reflexes is by the inferior colliculi. These structures are formed by migration of neuroblasts into marginal layer, where they organize a layered nuclei. So till now, this diagram summarizes brain development. That is, telencephalon forms the cerebral hemispheres. Telencephalon forms the cerebral hemisphere, and the cavity is lateral ventricle. The diencephalon forms the thalamus, hypothalamus, pituitary, and the cavity is third ventricle. The mesencephalon remains as a midbrain, and the rhombencephalon divides into pons, cerebellum, and medulla with continuous ventricular system. So, from vesicles to adult brain, structures and cavities evolve together. Now, coming to the development of the diencephalon. There are three forms, that is, three regions are there: lateral wall, roof plate, and the floor plate. Roof plate and the floor plate. The diencephalon can be understood based on its lateral wall, roof plate, and the floor plate derivatives. The lateral wall forms epithalamus. So you can see here, this is the lateral wall portion, epithalamus, thalamus, metathalamus, and hypothalamus. So this is hypothalamus. Epithalamus, metathalamus, thalamus, and hypothalamus. The roof plate forms the telachoroidea, choroid plexus, pineal body, and habenular nuclei. And the floor plate contributes to the hypothalamic structures such as mammillary body, and the tuber cinerarium, and is closely related to development of the pituitary via Rathke's pouch. We are going to see in detail development of pituitary as well. So this is about the development of the diencephalon. So the diencephalon shows a clear structural organization into roof wall and the floor and the derivatives with distinct function. Now coming to the development of the cerebral hemisphere, sulci, gyri, and lobes. As the cerebral cortex grows rapidly, it cannot expand uniformly within the skull, leading to folding of the surfaces. So you can see here the growth of the cerebral hemisphere. This results in the formation of sulci and gyri. So this results in the formation of sulci and gyri, which significantly increases the surface area for higher functions. Initially, primary sulci such as central and lateral sulci appear, followed by, so you can see here, this is the lateral sulcus, then this is the central sulcus. So initially these sulci are formed, followed by secondary and tertiary folds. These major sulci help to define the lobes of the brain into frontal lobe. So you can see here frontal lobe, parietal lobe, occipital lobe, and temporal lobe. With its structural development, there is also progressive functional specialization of different cortical regions. Now coming to the development of pituitary gland. The pituitary gland is an ectodermal derivative and develops from two distinct sources. The anterior pituitary, it is also called as adenohypophysis. So anterior pituitary, this is anterior lobe. Anterior pituitary or adenohypophysis arises from upgrowth of the ectoderm from the roof of the stomodium. Upgrowth of the ectoderm from the roof of the stomodium, which is known as Rathke's pouch or hypophyseal diverticulum. Whereas the posterior pituitary, in contrast, the posterior pituitary, or it is also called as neurohypophysis. So you can see, so this portion is stomodium, this is the Rathke's pouch, and you can see this is the anterior pituitary. It is forming by the Rathke's pouch. Whereas posterior pituitary or neurohypophysis develops from the downgrowth of the neuroectoderm from the diencephalon. So you can see here, from here it is growing downward. From here it is going downward. So neurohypophyseal diverticulum. These two components eventually come into close association with each other and forming the composite pituitary gland with dual origin and function. Now coming to the clinical correlation of the hypophyseal defects. Persistence of the Rathke's pouch will result into the sometimes forming the pharyngeal hypophysis. More importantly, these remnants can give rise to craniopharyngioma, which arises from the Rathke's pouch remnants, which are commonly located in the suprasellar region or along the pituitary stalk. suprasellar region or the pituitary stalk. Because of their location, they can compress the surrounding structure and leading to hydrocephalus. So these craniopharyngioma, which are in suprasellar or pituitary stalk region, results into the compression and results into the CSF pathway compression leading to the hydrocephalus. There may be possibility of the pituitary dysfunction leading to diabetes insipidus and growth failure. Another important malformation, that is Arnold-Chiari malformation. It is characterized by herniation of cerebellum through the foramen magnum. This leads to obstruction of again CSF flow leading to hydrocephalus. The underlying mechanism is tethering of the spinal cord due to abnormal development. As the vertebral column grows, this tethering exerts a downward pull causing cerebellum to herniate and will lead to the hydrocephalus. This can also lead to compression of the brain stem and other structures, and there are chances of CSF blockage. Clinically, hydrocephalus is managed by placing a ventriculoperitoneal shunt, which diverts the CSF to the peritoneal cavity. So this is about the Arnold-Chiari syndrome. Another cranial defects are holoprosencephaly. This holoprosencephaly represents a spectrum of development abnormalities resulting from failure of forebrain to divide into two hemispheres. So this leads to loss of midline structures affecting both the brain and the face. So in most severe form of this holoprosencephaly, which is known as allobar holoprosencephaly, the lateral ventricles merge into a single telencephalic vesicle. So it merges into the single telencephalic vesicle, and the cerebral hemispheres remain fused. These brain abnormalities are often associated with midline facial defects, such as fusion of the eyes. So you can see the midline facial defects in this diagram, fusion of the eyes and the single nasal cavity, single nasal cavity. This So the key concept is failure of forebrain cleavage leading to combined brain and facial abnormalities. So these are associated with the fused eye. The condition fused eye is called as cyclopia. Another anomaly, that is schizencephaly, rare cortical malformation characterized by presence of clefts in the cerebral hemisphere. These clefts typically extend from the ventricular surface to the cortical surface and are lined by gray matter, indicating a defect in neuronal migration. Depending on the severity, it can lead to loss of brain tissue and neurological deficits. So the key feature is full thickness cleft with abnormal cortical organization. So neuronal migration defect, that is schizencephaly. Last time also we have seen this anencephaly, which also comes in the neural tube defect due to failure of closure of the cranial neurophone, which must occur at the day 25 of the development. But this absence, this leads to absence of major portion of the brain, especially the forebrain, along with failure of formation of the cranial vault. So you can see absence of the brain and also the vault is absent. The exposed neural tissue undergoes degeneration, forming the area of cerebrovasculosa. Clinically, it is incompatible with life, making early prevention through folic acid supplementation extremely important. Another important anomaly, that is anencephaly with craniorachesis. In more severe cases of the anencephaly, the defect extends caudally, resulting into the craniorachesis, which there is failure of. So you can see there is a absence of vault, absence of brain, and it is extending downward. So failure of closure of entire neural tube. And this produces a continuous open defect involving both the brain and the spinal cord, resulting the most extreme form of neural tube defect. Both the conditions are incompatible with life, highlighting the importance of early neural tube closure and the folic acid supplementation. Hydrocephalus refers to abnormal accumulation of cerebrospinal fluid within the ventricular system, leading to ventricular dilatation. It may result from obstruction to CSF flow, that is non-communicating or impaired absorption, communicating hydrocephalus. The common developmental cause include aqueductal stenosis, and the malformations like Arnold Chiari we saw, which block the normal CSF pathway. This results or this leads to increased intracranial pressure and enlargement of the head in the infants due to open sutures. The management often involves diversion of the CSF using a ventriculoperitoneal shunt that we saw. And the last anomaly, that is microcephaly. It is a condition characterized by reduced head circumference due to impaired brain growth, particularly of the cerebral cortex. It often results in defective neurogenesis. Defective neurogenesis. Or it is neuronal proliferation. Neuronal proliferation during the early development. And these causes include genetic abnormalities, intrauterine infections such as torch, and exposure to teratogens. So these are the causes of microcephaly. Because here the skull follows brain growth, reduced brain size leads to the small cranial vault. Clinically, it is associated with intellectual disability, developmental delay, and also the neurological deficits. So these are my references. These concepts are primarily based on the standard embryological textbook I have taken, that is from Langman's Medical Embryology, and also Essentials of Human Embryology, 5th edition by A.K. Datta. So thank you all for your attention and participation. I hope this session helped in understanding brain development as a dynamic and clinically relevant rather than just a sequence of stages. If there are any questions, clarifications, or points you would like to discuss further, I would be happy to take them. Thank you.
Thank you, Dr. Sandeep Pakhale, sir, for your extensive and detailed lecture. Those who want to ask question can write in the chat box. Now I welcome Dr. Vatsala Swami ma'am, Dr. Gangne sir, Dr. Joshi sir, if they want to comment.
Good evening, everyone. Am I audible?
Yes, ma'am.
Yes,
Good
ma'am.
evening.
Good evening, Dr. Pakhale. I'm sorry I joined little late because I was in a meeting, but I think the postgraduates who have attended must have realize that this is really beneficial and advantageous for them, because the way you have explained the applied part of it, the applied importance or clinical anatomy, is the one which they have to apply to the basic knowledge that they have about the development. That's what I realized. It was very nicely explained. I was there for the later part of it, and I must appreciate the efforts put in by you. And I think the same is will be, I think, described or explained by my own senior colleagues who are there with us. God bless you, Dr. Pakhale. Very nicely described applied part
Thank
of
you,
it.
ma'am. Thank you, ma'am.
So, hello. Can you
Yes,
hear?
Yes, sir.
So, congratulations, Dr. Pakhale.
Thank you, sir. Thank you.
Very happy to see that the whole development of the brain has been narrated very briefly in 1 hour. That is the appreciable thing. Otherwise, the lectures which we required to take the brain develop, not less than that of the 10 lectures. But
Yes, sir.
you have simplified that in the 1 hour 10 minutes and focusing on the most important things which undergraduate as well as the postgraduate students should remember, and the details, if they want anything more, they should go through the books which are there. But no doubt you have explained it very nicely, the whole development. I have also I think joined at about 10 minutes only late, not more than that, to your lecture.
Yes.
Whole lecture, I had gone through your whole lecture, and really nice one and brief narration with good understanding to those who are attending this. Thank you.
Thank you, sir. Thank you so much, sir.
Hello.
Yes, sir.
Well, am I audible?
Yes, sir. Yes, sir.
Hearty congratulations. Already Dr. Vasla Swami Madam and Dr. Doshi sir have explained what you have done. It's a really wonderful lecture that you have delivered. Hearty congratulations. I'm sure it's going to be beneficial to all the postgraduates as well as revision for the faculty. Thank you. Thank you for nice lecture.
Yes, sir. Yes, sir. I have not covered the genetics part of it because I have to
No, it would
cover
have been
in.
very lengthy then.
Yes, sir.
And
Yes, sir.
you have summarized everything nicely. I think that should be enough.
Yes, sir. Yes, sir. Thank you, sir.
Dr. Kirti?
yes, ma'am.
Yes. I thought some any other faculty, senior faculty, or the faculty would like to add something more, because
Questions are there
it's
in
a
the chat box.
Any questions?
because it's
I
a very
just want
vast topic.
Yes, sir. Yes, ma'am. On chat box, there is no as such questions.
There are just appreciations by the faculty members for Dr. Pakhale sir's lecture.
Mm-hmm.
If any PG students want to ask any question, they are free to ask. Please unmute yourself and ask question.
Mahima Sharma, ma'am.
No, she was,
Okay,
there
okay.
was some background discussion.
Okay.
If any questions are there, they can ask me on WhatsApp also later on. And right now, if any questions are there, I can answer.
Just I wanted to ask about some neurobiotaxis.
Yes. Neurobiotaxis, it refers to the movement or migration of neurons or other processes towards the regions of greater functional activity or stimulation during development. So here neurons are not randomly positioned. They shifted towards the areas that provide stronger stimulation or synaptic input. So that can be classical examples. We can motor nuclei in the brain stems that I told,
Yes.
inferior olivary nucleus, and anything else?
Like we say, no news is good news. So no questions means it's very good.
Thank
Thank you. Thank you. If there are no
Yes.
questions, I will conclude this session. I would like to thank Mr. Tushar Kotwal for technical support. I really thankful for the Sandeep Pakhale sir for this extensive and detailed lecture. Thank you so much, sir.
Thank you.
Thank you all seniors.
Thank you, respected all senior faculty members. Keep blessing us.
Yes.
Thank
Thank
you,
you
sir.
all PGs.
Thank you. Thank you.
Hope to see you in the next session of RCA. Thank you all.
Thank you, sir.
Okay.
Thank you.
Thank you very much. Thank you.
Thank you, Kirti.
Thank you. Thank you, Rashmi.
Nice.
Thank you, Pakhale sir.
Thank you, madam.
Thank
sir.
you. Thank you, Kirti, for conducting
Moderating
it
this
so well.
session.
Sir, thank you.
Yes, for
Thank you
moderating.
for joining.
Rakhi ma'am also, I think she is traveling, so
She's,
couldn't join.
so she was unable to attend.
Bye. Over is there. See you in the next event.
Yes,
Yes,
yes, ma'am. Thank you.
Sure.
Thank you, everyone. Bye.